Good. So we'll start off. My understanding is that last week the webinar was all about how we collect data with the tractor out in the field, the logistics of the tractor design and setup. And so the first part of the talk will now discuss what do you do with the data? How do you take the data from the tractor and turn it into something that can be useful for analysis purposes? So we're going to be using geospatial data processing for that. First, I'll just review quickly what we have on board the tractor. We have three different types of sensors, including infrared thermometers, which measure the canopy temperature, an ultrasonic transducer, which measures the distance, basically, from the top of the canopy to the sensor, and that can give us an observation of plant height, and multispectral sensors, crop circles, which measure the reflectance of the canopy. We have one of these sensors on each of 4 rows on the machine, and all of the data that's collected by these sensors is geolocated by one GPS receiver. This shows a just basic kind of spreadsheet design of the experiment that we've used this machine and this instrumentation for. Just to review, we have several different cultivars of cotton growing in the field under both well-watered, WW means well-watered, and water-limited conditions. So this is just a not-to-scale spreadsheet version of the plot map. What I'm going to be talking about now is making this map and the data analysis true to the Earth by using geographic information systems. So these tools allow us to appropriately consider the geospatial nature of the data, and there are many GIS systems available. You may have heard of GRASS and Quantum GIS, which are 2 open source GIS software, and Esri ArcGIS is probably the most popular proprietary GIS. I'm particularly fond of Quantum GIS because it is open source. It's very lightweight. It can be easily downloaded for free offline and installed on multiple different operating systems. One of the things that makes it very useful is that it can be extended by developing custom plugins that you can use to automate your geoprocessing tasks in a way that is efficient and tailored specifically for your application. And so I've developed a tool which I'm calling HTP, for high throughput phenotyping, geoprocessor, which basically handles taking the data from the tractor and turning it into a useful data format for further analysis. So the HTP geoprocessor is basically an open source plugin that can be installed within the Quantum GIS environment. It's important to note that this processing happens independent of the data collection in the field. So the tractor is collecting a data stream, saving that to a file, and then these files are further processed in the computer lab. One of the design criteria for this software, since we have data coming from 3 different types of sensors, which ended up providing 3 different file formats, is I wanted a tool that could operate independently of the particular file structure for a particular sensor type. So this is a basic layout or screenshot of the Quantum GIS environment, and I'm highlighting this plugins menu here with my tool installed. And the tool does 2 basic tasks. We pre-process the data, and then we geoprocess the data in relation to our plot boundaries. Shown in the map view is the actual map of the field experiment that I introduced earlier. And if we zoom in on a part of the field here, we can see our very long rectangular plots representing 1 row of cotton. Each rectangle represents 1 row of cotton. And we have some sensor data points that were collected with the tractor overlaid on top. This is sort of the final product, the data points here. as they are located as the final product from this software that I will be describing. So the geoprocessing tool performs two basic tasks: pre-processing, which reads the sensor data from the file. Again, this is independent of the file structure. We convert all the latitude, longitude coordinates to the Universal Transverse Mercator, or UTM, coordinate system. And then we calculate coordinate transformations from the GPS receiver position to the position of each sensor along the boom. Of course, this depends on the vehicle travel direction through the field and also the geometry of the sensor in relation to the GPS receiver on board the tractor. And secondly, the geoprocessor is used to analyze the sensor data iteratively within the plot boundaries. So we can do things like calculate summary statistics for the data within each plot. We can assign a plot ID number to each sensor data point. And this is very important for the analysis that Mike will be describing next. Of course, this requires that an accurate plot boundary map be created and loaded as a polygon shapefile within the GIS system. So first, pre-processing. And the first step is to read the data. So as I mentioned, the tool must read data independently of the sensor type. And the reason is generally the tractor collects data from each sensor in a set of 4, but the format for the way that it saves the data to a file is different. For a crop circle file, it might save the sensor data down the row, so each row has a different piece of sensor information. For, say, the sonar data, it would save that group of 4 sensors across the columns. So you have different pieces of data across the columns, and we need a tool that can work with either type. So this is how we specify, we tell the GIS how to interpret or read a particular sensor data file. The tool expects a comma delimited file. We basically specify the total number of columns. We list our sensor names here. In this case, we have 4 sensors. The latitude column is always the second column. Could be different columns, but here, for this particular case, the latitude always appears in the second column, longitude in the first column, heading information in the 7th column. If there's a sensor ID, it's located in the 8th column by this particular setup, and a sensor ID text is defined. For the case that we have sensors located in rows, different rows, we need an ID to tell us which sensor this piece of data is coming from. The first column containing data that we're interested from a sensor is specified here in column 9, and then this entry tells us how many columns of data there are per sensor consecutively. And importantly, we have to enter the easting and northing offsets in meters. This basically assumes that the tractor is facing a due north position, and we have to enter the offset position or distance from the GPS receiver to the position of each individual sensor. So by running this tool, we can get the data from the sensor file loaded into the GIS. Next, the tool converts latitude, longitude to UTM coordinates. So all GPS receivers provide information in latitude and longitude in terms of degrees from the equator for latitude and degrees from the prime meridian for case of longitude, and also an elevation measurement in meters. GPS receivers are based on a WGS 84 datum. This is basically a set of data that describes the shape of the Earth. Because the Earth is spinning, it is not a perfect sphere, and so the reference ellipsoid tells us about the shape of the Earth and also the shape of the oceans from the geoid due to the rotation of the Earth. This information is then used to calculate a map projection. So we're converting the latitude and longitude to a Cartesian coordinate system so we can measure our distances between points in meters. This allows us to use simple trigonometry to treat the geospatial data by Euclidean distance. If you remember from your geography classes, you will have some distortion when you perform these map projections, but at the scale that we're working for high throughput phenotyping, we expect this to be minimal. This slide demonstrates why it might be important to convert to UTM coordinates. So I have a diagram of our vehicle here, where I'm positioning the vehicle at an easting of 4 and a northing of 4. Okay, the GPS receiver is denoted by the red dot. So we can see from the two sides here that the GPS position is in the same location. However, because of the orientation of the vehicle, we have different positions for each of the sensors. So we have to calculate the sensor position based on the GPS position, the heading of the vehicle, and the known distances from the sensor to from the GPS receiver to each sensor. And so the pre-processing tool will accomplish that task. So once that's done, then we can load the data files into the GIS, and we can overlay that data onto a plot boundary map. So we see here that the tool has accurately loaded the data and computed the sensor data positions. For this case, the tractor would have started here in the southeast corner, driven to the west. So we have 4 rows that the tractor is sampling. It would have turned around and sampled the next 4 rows, and on through the field. And this then shows what data is contained within the attribute table for the sensor data shapefile that we would have created. We have our 4 sensors that are grouped by their latitude. So the latitude is all the same for a set of 4. Heading is all the same. We've calculated the UTM position for the GPS receiver. We then calculated out to each of the 4 sensor positions, and we noticed that the UTM sensor positions are different for each sensor. And then on out here we have the data. Now the attribute table for our plot boundary shapefile, this was created with other GIS tools that are already contained in Quantum GIS. We might have some information of interest about a particular plot, including, say, the block number, the treatment number, the rep number. But the most important thing is the barcode. So each plot in the field was assigned a unique barcode. And so what we want to do with our GIS processing is to assign the barcode of a particular plot to each sensor data point that falls within it. Then we can always tie a particular sensor data point to the particular treatment that was administered at that location. So to do that, we have a second tool called the geoprocessing tool, or the vector geoprocessing tool. And we can do a couple things with this particular tool. It's basically for just analyzing the sensor data points in relation to the plot boundaries. So we can calculate, say, the mean value or other statistics of points that fall within a particular polygon, and we can assign that barcode to from a polygon to each sensor data point. And then to wrap up, we get this final result where we have our UTM sensor data locations, and we have the 5 for this particular sensor. We have 5 data values that we're interested in from the sensor at that particular location, and we have assigned the barcode for that particular plot, so we know exactly what treatment is imposed at that location. And this is the sort of data that I would hand over to Mike for the analysis that he will talk about now. So I'll turn it over to Mike.
Okay, so my name is Michael Gore, and I'm an associate professor in the plant breeding and genetics department at Cornell University. And I thank everyone for joining today. And I thank Sean Yarns for the introduction and John McQueen for helping out with technology. So what I'm going to be talking about in this section is about processing all of this phenotypic data that's now been collected by the high-throughput phenotyping tractor platform, and then how to properly analyze it. So here we have in an Excel format layout a geoprocessed raw data. So this is coming out of Kelly's geoprocessing computer package that he created. And so now there was some post reformatting, so it would be compatible with SAS or like R software packages. Ooh, whoops. There we go. So, and one of the most important things about phenotypic data, or just quantitative data in particular, is you need to know your data. And the best way to do that is with a histogram, a box plot, or a scatter plot, QQ plot. So the quantile plot is a graphical method for comparing two probability distributions to assess goodness of fit, and generating a bunch of summary stats such as mean, standard deviation, variance with their confidence intervals, looking at the normality of your phenotypic data, testing for outliers. You can also do quantiles, and you can generate their and look at their related confidence intervals, and you can do frequency counts for observations. So as like what Kelly was showing on a row by row or a plot by plot, then you can actually, quantify how many readings are there per plot. So at the speed for which the tractor was traveling, on average, there were maybe 8 to 10 readings or like points per plot, and each plot can consist of anywhere, depending on the actual stand, from like 50 to maybe 80 cotton plants. And you can also look at or create plots to see how much phenotypic values are not present. So say if you had like a failure in one of the rows, you could then look to see if maybe some of the plots have like no phenotypic information at all, or maybe have like tenfold less phenotypic values. So that's important information to know. So I really like this quote from Parnett and Lewis, and not all outliers are illegitimate contaminants, and not all illegitimate scores show up as outliers. So the main point I'd like to convey from this is outliers are unusual data points that substantially deviate from the mean and strongly influence parameter estimates. You should always check for outliers in your data sets. If you're getting any phenotypic data sets from a collaborator or some other lab, this is one of the first things that you should do. You should always check for phenotypic outliers and then generate like the summary stats so you have an idea of the quality of your phenotypic data. And if there are outliers detected, do not ignore them. And the reason why is because this can result in several negative consequences, such as an increase in error variance, reducing power of your actual hypothesis testing, distorting estimates of your parameters, decreasing normality if non-randomly distributed. And one important point I'm stressing here is that inferences made from these fundamentals may not be representative of the population under study. So in a sense, this is saying your conclusions or inferences that you're making from your results may not accurately reflect the population. So what are some of the potential causes of phenotypic outliers? Human errors, so this could be in phenotypic data collection or the computational code that was written to process all of the phenotypes. So this could be recording, say, with the tractor or with a Palm Pilot, or inputting manually into like a computer like spreadsheets such as Excel. So one thing that's really powerful with this tractor approach is we're not having to do any like manual imputation at all because everything is collected on the tractor and then it's geoprocessed. There can be technical errors from faulty or non-calibrated equipment used for phenotyping, so it's imperative at the beginning of every season and even partway through the season to actually investigate the quality of your equipment just to make sure that it is not faulty and to make sure it is calibrated. And so there could also be sampling errors because of different subpopulations, incorrect assumptions about temporal trends. And one thing to also keep in mind is some of these biological outliers could be actually real because there is a 1% chance of getting an outlier that is 3 standard deviations from the mean. And also there could be large gaps in the vegetation. And this is one of the major causes of phenotypic outliers in our phenotyping data sets, because when there's poor stand count, there's large gaps in the vegetation, then the soil is exposed. So in a sense, that soil is contributing noise and contamination to the phenotyping when it's actually being carried out in the field. So it's imperative to remove that noise, or your as I was saying earlier, inferences and your hypothesis testing may not be valid. So how do we detect these phenotypic outliers? So one approach that I've used in the high throughput phenotyping data sets, and others that have also used in large phenotypic data sets from maize and many other plant species, is fitting an initial linear model of fixed effects for each trait. So, possible fixed effects to include in the model are such as cultivar or line, or it could also be called a genotype. And then if there's a treatment effect, such as like drought or, wet or dry treatments, in environment terms, so you may be doing like 2 or 3 locations of testing of your same genotypes of germplasm, and you may have reps. Your like calendar year and the actual time when you're actually going out to the field and doing the phenotyping. So because if you're doing a trait such as like canopy temperature, it'll change over time. So this is a covariant you may want to also put in your model to look for outliers. And sometimes and often you can test for the interactions of these terms as well when you're searching for outliers. So some of the possible approaches to test for outliers I have listed here. Now there are one thing I just really want to be clear here. There are several possible approaches, and I'm not advocating any one. So if you have questions, you should maybe consult with people who are experts in this area. But I will say that these 3 are ones that work very well for my research. And in a sense, what you're doing here is you're testing for the influence and the leverage of a particular phenotypic value. And then if it's great at a certain like cutoff threshold, then it needs to be removed. And one important aspect of the detection of the phenotypic outliers is you need to repeat model fitting several times, because as you remove outliers that had maybe great leverage or great influence, there may have been other outliers that were masked, if you will. So one important aspect that folks often don't consider when they're analyzing phenotypes is often the traits are non-normally distributed. So when fitting linear models, 2 very important assumptions are that the error terms follow a normal distribution and there's a constant variance. So when your phenotypic data are non-normal, these 2 assumptions in particular could be violated. So you can use a generalized linear mixed model to analyze non-normal phenotypic data, or you can use the Beacock's procedure, And how this works is you find the most appropriate transformation that corrects for non-normality of the error terms and unequal variances. And I have a reference here for this transformation procedure. And on this next slide is some of the more common transformation procedures that you could use. And this procedure can either be carried out in, say, SAS or the R programming language. And how this, if you will, how this algorithm works, it is looking for the most optimal transformation for your phenotypic information. Now, just remember, after you transform something and you want to report it in a table, you will in a sense need to reverse transform it to put it back in its same values. So this is a QQ plot. So as we see here, this is comparing the normal theoretical quantiles and the expected quantiles. And what we see here is that there is tremendous skewing in this phenotypic trait here. But after we do the outlier removal procedure and we use a Bloch-Cox transformation procedure, we can then see that now this is following the expectations that we would want for our phenotypic data. And now this can be used with any general mixed model procedure to analyze the phenotypic data. So let's talk for a little about mixed linear models here. Here we have y as a vector of phenotypic observations. Then we have a vector of fixed effects, and u is a vector of random effects, and e is a vector of error terms with mean vector and then variance. And that's in a sense capturing the residual variance. And then your x and z's are incidence of ones and zeros, and you're relating your y to your various vector of fixed and random effects. So that's just like a very brief general overview of the testing structure of a mixed linear model, which most of you have probably already come in contact with in your classes. So with a mixed linear model, you can either calculate blues of fixed effects. So a blue is a best linear unbiased estimator, and that's generated for fixed effects. And then there are BLUPs, which are best linear unbiased predictors, and those are only estimated for random effects. And all of this is in the framework of a mixed model, and this can be carried out in, say, such software programs as SAS, R, or ASRIML. And so our first mixed model that we have here is an example of accounting for field effects. So what we're trying to do here is predict BLUPs for each line for a particular phenotype. So if you look at the random terms in this model, now the only fixed term in this model is the grand mean, but all of the other terms are random. So we have a term for environment. So also sometimes there could be, depending on the size of your field test, it could be broken across like 2 or 3 fields. And so we have a field effect there. We have a rep effect. Since this is an incomplete block design, we have a block effect. And something we also do is we account for like the grid pattern in the field, so accounting for row and column. So that helps take, if you will, takes into account some of the environmental variation that may happen in the field that could be caused by like soil. And we have a random error term for line, and then an environment by line interaction. So we could also then do this for, in a sense, blues for fixed effects, and then in the SAS programming language, we can have at least square means, and that's obtained for fixed effects. And in this particular model, we're very interested in the actual like F value, if you will. So we're testing the importance of line treatment and line by treatment interaction in relation to our phenotype. And rep and treatment and block and row and column, those are actually random terms in the model. So after we remove our outliers, transformed our phenotypic data, so what I was saying earlier is generally there's like 8 or 9 readings per plot. So then what we have been carrying out then is we take a mean of those, say, 8 to 9 readings per plot, and then we analyze it with a weight mix model, and then using the number of observations per plot as, if you will, a covariate in the mixed model. So this seems to work fairly well when you have a very small population size, but when you have a very large population size, say of like thousands of genotypes, there's often issues where the mixed model will not converge, and so if that's the case, you may not be able to, in a sense, use this covariate in the mixed model. But this is a possible approach that should be tried first. And then, so with this phenotyping system, it can vary across space and time. So let's talk first about space. So we can fit a separate spatially autoregressive correlation structure in the row and column directions. So this is, in a sense, looking at the AR1 error structure for each environment. So this is like row by column. So it's the various row by column combinations being tested across the field. And then you can also model the correlation structure for repeated measurements of a trait on the same experimental plots over time with a variance covariance matrix. And how this works is because it's the same genotypes and the same plants that are being repeatedly measured across a particular time point for the whole growing season, or even across like 2 or 3 time points across the whole growing season, then those repeated measurements of a particular trait on the same experimental plots are going to have a correlated structure. So it's important to account for that in the mixed model. And after like all these terms are included, then you can actually do a likelihood ratio test to remove terms from the model that should not be in the model at an alpha of 0.05. And then the final model is used to estimate BLUPs for each line and to estimate variance components. So one way to look at the quality of your phenotypic data that's being collected is to calculate, say, the broad sense heritability on an entry mean level, or it can also be referred to as repeatability. So in a sense, you want to know what is the proportion of the phenotypic variation that is attributed to genes, i.e. quantitative trait loci in a particular genome or in a particular population. And to calculate the variance components, you can use the same mixed models that we talked about previous, but all of the terms need to be random so that you can derive a variance component. So if your heritability is high, then that can then tell you there is a large amount of the phenotypic variation that's observed in a population that is attributable to genes. But if your heritability is low, then a large amount of the variation that you're seeing for a particular phenotype is attributable possibly mostly to the environment, and that's something you do not want as a plant breeder. And then with these BLUPs, you can also estimate phenotypic and genotypic correlations with a mixed model that in a sense can be a multivariate model where you're including 2 or more traits at the same time to see what the genotypic correlations are. But generally, I just do pairwise. And then with the approach, you can calculate the standard errors of all these heritabilities and the correlations as well. And I've included a reference that has a link to all of the SAS code where all these calculations can be followed and carried out. So after you have processed your phenotypes, you've removed all your outliers, transformed them, and generated either blues or blups, that's when you can begin to really understand the genetic architecture of your phenotypes. And there's various approaches to carry that out, or if you want to carry out this work in a context of a plant breeding program. Now, I included more papers on these topics for further reading because each of these approaches I could spend an hour on for each one. So for the main ones, it would be like QTL mapping. So that can be either one trait or two or more traits that are correlated at the same time. And the same thing for GWAS. So that's just a different type of unrelated population design. We could do GS, so this is in the framework of a plant breeding program where you have a training set and you're actually trying to predict certain phenotypes from the genotypic information. And one area that I think is growing and is going to be actually very useful is being able to integrate plant growth models into these QTL approaches so that we're able to actually map QTLs in reference to the plant growth model over time for the whole growing season. So this is now shifting gears where I'm just going to give an example of the application of this work. And this is a recombinant inbred line population of cotton. So about 100 recombinant inbred lines, and it's hirsutum. And we have two treatments, wet and dry, and 2 reps. So if you follow my cursor here we have dry rep 1, wet rep 1, dry rep 2, and wet rep 2. And you can see the 100 or so plots for dry rep 1. You can see it's very like kind of like a light greenish color compared to this like royal blue color. So the greener it is, the hotter it is, and the bluer it is, the cooler it is. And so what we're able to do here, so this is all collected with the high throughput phenotyping tractor with the IRTs, so we're able to recreate a heat map of the field. And I'd like to call your attention to this one area of the field here. This area of the field actually had a higher clay content. So where I was talking about including an autoregressive model with row and column, so that's to help to account for some of the environmental induced variation that is, causing the actual phenotypes to change. And this is, if you will, a proof of concept. So here we have the red line, which is the population mean for the 100 recombinant inbred lines, and this is for the drought, the dry treatment, and this is canopy temperature. So the x-axis here is hour, and it ranges from 10 a.m. to 1 p.m. And on the y-axis is the canopy temperature in degrees Celsius. So this blue line here is the wet plots. So those are our control plots, and you can tell that the canopy temperature is much cooler on average for the population mean, and the black line here is the ambient air. So there is a treatment effect, a time effect, and a time by treatment effect. And this was tested in a mixed model framework. And these were our fixed effects. So the next question is, okay, we know that there's phenotypic variation, but is it heritable? Is it under the control of QTL, i.e. genes? And that's what we're calculating here. We're calculating the broad sense heritability for the plots that are wet. So those are control plots, which have a mean of 32 degrees Celsius. And that had a heritability of 0.52, and then the red colored plots, if you look over here, they had a mean of 37.98 and it had a heritability of 0.8. So these traits are heritable, and what that says is that if plant breeders wish to use this phenotype in a breeding program, it could be a possible tool. And now we wanted to link this with genotyping by sequencing in QTL analysis. So what this is for those phenotypes that I showed you, this is taking the ratio of dry to wet canopy temperature, and this is over a period of 2 weeks at 7 a.m., 1 p.m., 7 a.m., 10 a.m., and at 1 p.m. So up here, we have the chromosome position of a particular marker, and the blue here means that the QTL effect coming from the one parent TM1 will decrease the canopy temperature, whereas the red color QTLs coming from the parent TM1 will increase the canopy temperature. So what we draw from this is that over these 2 weeks and at these various time points, the QTLs seem to be acting in the same way. That is, they're always going to either decrease or increase the canopy temperature in the same fashion, but they don't have their strongest expression until 1 p.m. for each week. So that shows us that the QTLs are like turned on the strongest at 1 p.m. each week, and that happens to correspond with like the peak heat stress and drought stress for this population. So that just puts into practice kind of what Kelly showed with the geoprocessing and what I showed with trying to remove all the phenotypic outliers and transform it, and then to proceed with QTL mapping. And now Kelly is going to talk about inverse modeling. So this is taking it even further from QTL mapping.
Okay, thanks, Mike. Just to wrap up, I would like to talk a little bit about inverse modeling. For those of you that don't quite understand what that is, I'm going to provide a basic introduction to it. But what we're essentially trying to do is extend the amount of useful information that we can use for the data analysis that Mike described. Our tractor is really, at the core of it, only collecting 3 pieces of information. We have the reflectance of radiation from the canopy, the temperature of the canopy, and the essentially distance from the canopy to a sensor, which is a surrogate for crop height. So how can we use our models, our physical and ecophysiological models, to estimate additional traits that can be used for phenotypic data analysis? And the answer may be using some kind of inverse modeling procedure. Okay, so here is a basic example of inverse modeling. Typically when a modeler is designing a model, the way that it's done is attributes of a particular system are identified. And those are the inputs to a model. And then the model is programmed to simulate some quantity or some value that can be observed about the system. So ProSail is an example model that's used quite often. It's a radiative transfer model used in remote sensing. This model basically takes as input things like the chlorophyll A and B content of a leaf, the water content of a leaf, the leaf area index of a canopy, and a few other parameters. And the model will then simulate what the canopy spectral reflectance response should be. And for typical vegetation, we get some response like this, where the wavelength here is 400 nanometers and out here is about 2,500 nanometers. So if we were going to do a forward modeling procedure, we would basically know all of these inputs. These various inputs about the canopy would be known. We would run the model forward and get the output. So very simple. But that's typically not the case, and it's not the case for phenotyping. For phenotyping, we're measuring reflectance. We're measuring what the model outputs. And so then what we need to do is use that information along with the model to estimate what the model input should be. So if a model can be mathematically inverted, then we can calculate that directly. However, most models used in the physical sciences are not able to be inverted because they are complex. And for that reason, we have to turn to optimization, which essentially means running this model forward many times until we find the solution or the input parameters that minimize the error between the observed and simulated model output. When we do optimization, that brings about a whole host of other questions that need to be addressed, such as which model parameters do you adjust, which ones do you constrain, for the ones that you adjust, what bounds do you allow them to be flexible within. For the case of ProSail, what spectral wavelengths do you include in the optimization? When do you observe the canopy, at what time? And what objective function are we minimizing or maximizing? Depending on the decision for all of these questions, the outcome of the optimization will be affected, and so we need to very carefully address each one of these. So my basic methodology to test this approach has been to use the data that we have collected in the field. Again, this is for our cotton experiments. We have 3 years of cultivar by water experiments. And I have gone out to the field and collected hyperspectral reflectance information of the crop canopy. This is actually separate from the tractor because this instrument is much more expensive, and we can only afford to have it carried in the field. So I've carried this instrument in the field and collected spectral reflectance responses of the canopy. I've then taken that data back into the computer lab and built a computational program that implements ProSail. It implements an optimization algorithm called PEST and uses Python for basic control to conduct these inverse modeling or optimization experiments. So the basic procedure has been to first evaluate those questions that I asked in the previous slide and try to figure out what is the best protocol for the optimization. Then we can do things like compare measured and simulated results to make sure the model is performing adequately. And importantly, we can use statistical tests to test the significance of optimized model parameters, which essentially are other traits that we're observing through this exercise of our canopy. So here again is a little bit of a summary of our field experiment. We have 3 years of data from 2010 to 2012, well-watered and water-limited irrigation on cotton and many different cotton cultivars. I'm going to be focusing here on observations collected in the AM and PM on August 4th and in the AM on August 18th, 2011, 2 different years. The instrument that I'm using is an ASD Field Spec Pro, and it essentially measures reflectance of light between 350 and 2500 nanometers at a 1 nanometer bandwidth. This is a picture of us using a spectralon panel to correct that data against the incoming solar irradiance at the time of measurement. We also have some other data to go along with our spectral measurements, including leaf water content, specific leaf area, and chlorophyll A and B content. A little more about the ProSail model. ProSail is a combination of Prospect, which is a model of the way that light interacts within a plant leaf, how the plant leaf structure treats light, reflects it, absorbs it, and transmits it. And the sail model, which is essentially a model of how solar angle, solar geometry affects how light is reflected from a plant canopy. 14 input parameters are required that describe leaf pigment, leaf water content, canopy architecture parameters, solar geometry parameters, and some others. And then as an output from this model, we get the canopy bidirectional reflectance from 400 to 2500 nanometers in 1 nanometer increments. So the output of this model corresponds very nicely with the observations that we're collecting with our hyperspectral equipment. So to parameterize the model, I'm holding several parameters fixed. Some other parameters are based on observations in the field. And the key thing to notice is that I'm going to be optimizing 3 parameters that are basically an N parameter, which is a structural parameter of the canopy. LAI, leaf area index, and the leaf inclination angle will be optimized to minimize error between our observed spectral data and our simulated data. So this diagram kind of shows the procedure that we're doing. We implement our pest optimization routine. Into that goes the output spectral response from ProSail and the observed spectral response from our spectroradiometer in the field. We then use the optimization to intelligently adjust these 3 parameters that we're optimizing. And then this happens many, many times until we come to a solution for the optimization where we've adjusted these 3 parameters to minimize the error between the measured and simulated canopy spectral reflectance. And we're going to be focusing here on 3 different groups of bands: 400 to 1300 nanometers, 1450 to 1770, getting into the shortwave infrared. 400 to 1300 is more in the visible NIR, and 1970 to 2400 nanometers is on out into the further into the shortwave infrared. Then what we can do is some very routine ANOVA and multiple comparisons tests on our resulting trait estimates that we're getting through our model optimization. Here I'm showing statistically significant differences between LAI, between our well-watered and water-limited conditions for the August 4th date at 2010 a.m. And we have similarly some statistical differences among 4 cultivars. That's important because essentially that's what we're trying to do here, is find statistical differences among the cultivars and that information, the traits then that we're estimating, can be used in further genetic analysis that Mike described. One of the issues, if I go back, I want you to notice, is that the LAI in the morning is about 4 to 5. In the afternoon, on the same day, we're getting an optimized LAI of 6 or more. Of course, that is not realistic because we don't expect the canopy to grow that much in an afternoon. So this highlights one issue with optimization, is that you have to be very careful about the values that you're getting, making sure that they are realistic. Again, changing the protocol, the procedure for the optimization can highly affect what values we actually get. The important thing to notice is that we're getting statistically significant differences from these trait estimates among our treatments in the field. For the PM, we actually have now statistically significant differences between our leaf angle, probably due to the increased leaf droop due to water stress in the afternoon. And on into the second year, we're again seeing more statistically significant differences. Okay, so in conclusion, we can maybe use inverse modeling to find statistically significant differences between trait estimates that are more difficult to observe in the field. And that's where the modeling comes into play. However, the results are highly dependent on how you set up the optimization routine. Plant growth models were mentioned earlier. I think plant growth models could also help in this case. If we know the temporal time series of a particular, say, LAI, a state variable in a model, that might help constrain the way that ProSail is estimating LAI through inverse modeling. So with that, I'll conclude and turn it over to Sean. Thank you.